Cell walls are found in plants, fungus, and many protists. Now the cell walls surround the plasma membrane, and they form sort of another layer. So if you look here at this diagram, on the bottom left it's labeled CW, and then there's two double arrows. That is the cell wall. Just on the inside of that cell wall would be the plasma membrane. The plant cell walls are made up mostly of cellulose, and fungus cell walls are made up mostly of chitin. Inside the cell walls is cytoplasm, which is a viscous fluid that contains the organelles. Cytoplasm is mostly made up of interconnected filaments and fibers, a fluid or cytosol, organelles, but not the nucleus, and storage substances. The cytoskeleton is made up of filaments and fibers, those three types are microfilaments, microtubules, and intermediate filaments. And they have three functions, which are mechanical support, to anchor organelles, and to help move substances. And if you look at this drawing, I have the three different types of filaments labeled. We have actin in the bottom, intermediate filament in the middle, and microtubules at the top. Okay, now cells also need to move. Cells move using cilia and flagella. Cilia are short and they're used to move substances outside of human cells. And flagella are whip-like extensions. Whip-like extensions that are found on sperm cells and basal bodies like centrioles. So here is the structure of cilia and flagella. You have an outer pair of 9 outer pairs of microtubules. You have inner microtubules, and you have a membrane. And there's a picture, a microscopic picture, and if you notice the label, they put 70,000x, so that's the magnification that they are looking under. Okay, now I'm going to skip ahead here, because we're not going to talk about all of these. Okay, what I do want to take a look at is a comparison of bacterial, animal, and plant cells. Now, if you have a question about any of these specific organelles, the information is all going to be in Moodle so that you can refer back to it, or you can refer to your biology book. But I want to look at what's different between the three main types of cells. So first we'll look at the exterior, and we have a cell wall. In bacterium, the cell wall is present and it's made of a protein and polysaccharide. In animal cell, there is no cell wall. And in a plant cell, there is a cell wall, but it's made of cellulose. And the plasma membrane is present in all three. And ribosomes are present in all three. Other than that, there is nothing that is either present or absent in all of them. So go ahead and just take a look at this chart that compares bacterium, animal, and plant cells. So moving in and out of a cell, when we were talking about the plasma membranes, there's four types of main movement: passive transport, active transport, endocytosis, and exocytosis. Passive transport is the easiest. There is no energy required for passive transport. It just happens. And it happens because of a gradient. That gradient could be a difference in concentration, it could be a difference in pressure, or it could be a difference in charge, meaning positively charged and negatively charged. And the movement occurs to equalize the gradient. The three types of passive transport are diffusion, osmosis, and facilitated diffusion. Molecules move to equalize the concentration. In this diagram, it's showing a lump of sugar being dropped into a warm beaker of water. Eventually, the lump of sugar is going to dissolve, and the concentration, the little orange dots, are the same throughout the entire beaker. This is diffusion. The molecules are moving by themselves to equalize the concentration. Another easy example of this is if you take a drop of food coloring and you drop it into a cup of water. The food coloring disperses or moves to make the entire cup of water the same color. That's an example of diffusion. Osmosis is a special form of diffusion. In osmosis, the fluid flows from lower solute concentration to higher solute concentration. This often involves the movement of water, either into or out of the cell. And there's a couple of different ways that solutions can be categorized. They can be hypotonic. In a hypotonic cell, you have solutes in the cell more or a higher concentration than on the outside of the cell. So the outside solvent flows into the cell. In an isotonic cell, you have the solutes being equal on the inside and outside of the cell. In the hypertonic, the solutes are greater on the outside of the cell, so the fluid flows out of the cell. Ideally, cells are isotonic. They like to be equal. And here's a diagram of hypertonic, isotonic, and hypotonic cells. If you notice, the hypotonic cell, if it gets too big, will actually explode. And the hypertonic cells can shrivel up. So if you have a plant that is wilting, its cells, the plant cells, are hypertonic. Facilitated diffusion is when the membrane is differentially permeable, which means there are specific channels that help certain molecules or ions either enter or leave the cell. These channels usually are transport proteins that facilitate the movement of water, and no energy is used during facilitated diffusion. So here's a picture of how facilitated transport takes place. You have protein binding with a molecule. The shape of the protein will change, and the molecule moves across the membrane. The high concentration is on the outside of the cell, and the low concentration is on the inside of the cell. In active transport, you do need energy because the molecules are moving against the gradient, or they're moving in the opposite direction. An example of an active transport is a sodium-potassium pump. And these are the 3 steps to the sodium-potassium pump. There's a sodium-potassium pump embedded in the cell membrane. There's potassium on the outside of the cell, sodium on the inside of the cell. And in a sodium-potassium pump, the sodium from inside gets pumped to the outside, and the potassium from the outside gets pumped to the inside. Endocytosis is moving movement of a large amount of materials, like particles, organisms, or large molecules. And the movement is into the cell. There are 2 types of endocytosis. There's a bulk phase, which is nonspecific, so it's not looking for a particular type of molecule. And there's receptor-mediated, which is a very specific molecule. And here's a picture of endocytosis. You have in step 1, the plasma membrane surrounding the material. In step 2, the membranes meet. And in step 3, the membranes fuse to form a vesicle. Endocytosis has 2 forms. It has phagocytosis, which is the cell eating, essentially, and penocytosis, which is the cell drinking. And then there is exocytosis. And exocytosis is the cell discharging material. It is the reverse of endocytosis. Exocytosis, the vesicle moves to the cell surface in step 1. Then the membranes of the vesicle fuse to the outside membrane, where the membrane is then expelled. So on the left you have a diagram, and on the right you have a still photo of the instant when exocytosis took place. And that is the end of cells. Now I can assure you that this class was definitely the most boring of these learning labs that we will have. But it is all information that I needed to make sure you guys had in order to complete the rest of their learning lab. So please don't get off today and think, oh my goodness, all she does is talk. This lab is going to stink. And let me assure you that it only gets better from here. So, that was our brief overview of cells. Now, like I said, there is lots of cell information I'm going to put up in Moodle, and there is lots of cell information in your biology books. What I would like to show you now is your homework. Up on the PowerPoint screen, under Documents, is your homework assignment. Now, it is listed in Moodle. You are going to create a model of a plant or an animal cell. This can be a 3D model that you take a photograph of. It can be a virtual model that you make on the computer. Or it can be a model that you practice your scientific drawing skills with. It is completely up to you. I would like you to be creative, and I would like you to use whatever skills you have. For example, I would not be drawing one. I know that my skills as an artist are very lacking. I would probably be making a 3D model out of something. I have had students make a 3D model out of food objects. One student used, I believe, a cantaloupe one year. I have had students make a 3D model using potatoes, using Legos or Kinects, using a basketball, whatever you would like. Or if you are not creative in that sense and you would like to just draw, go ahead. Draw on the computer, draw on a sheet of paper, whatever you would like. Along with the model, you are going to explain the model in a few paragraphs. By explain the model, I mean, let's say you choose to do a plant cell. You're going to do a typical plant cell. So you are going to have a cell wall, and you are going to tell me what your cell wall is. If you're drawing, the cell wall is the dark purple line across the outside of my plant. In a plant, the cell wall is responsible for what a cell wall does. It's responsible for the structure of the plant, holding the materials in. You also need to have a key to your organelles. You do not need to have every single organelle. It just needs to be representative. So your cell should have a nucleus. It should have ribosomes. It should have endoplasmic reticulum. It should have mitochondria. If you do a plant cell, it should have chloroplasts. And you also need the description in your paragraphs. So you have a lot of artistic liberty in this activity. We will do the same thing we did today. You will share whatever you have. So if you make a 3D model, you're going to need to submit a picture to me, as well as your paragraphs and your key, but then save your 3D model if it's possible. I mean, if you make it out of food and it rots, don't save it. But if you make it out of Legos and next Wednesday we can turn your cameras on and we can all see the Lego right in front of us, that would be a lot better than just being able to see the picture. This assignment is due next Wednesday by 9 a.m. so that I have time to check it beforehand. Your resubmission of your lab reports are due by 9 a.m. as well next Wednesday. Does anyone have any questions about the homework assignment or redoing the lab reports or anything that we talked about today? I would say if you have 10 organelles in your model, that is a good number to have. The key should be either written in your paragraphs or you can just list it on the side, and it should just tell me what everything is. So let's say you make a model, you draw it, and you have, let me just sketch something real quick here. So you have your cell, now again, I'm doing this very quickly. Yours will need to be much neater than this. But I'm looking for something, you know, here's my cell, and next to it, I have my key. I have that the starburst is the nucleus, the cell membrane is a straight line, and the small little circles are the ribosomes. So you can either make a separate key like that, and you even should label it key, right along the side, or you can write it in paragraph form. So you could write, the starburst is the nucleus is the control center of my cell. Either way is fine. Great question, Sarah. Anyone else have any other questions? All right, well, that is the end of class. Sarah, if you could stay on for 1 minute, that would be fantastic. The rest of you, I will see you all next week. Any questions between now and then, please go ahead and let me know. Either email me or give me a call. I hope you guys have a fantastic week, and I will see you next Wednesday.